Hello everyone, grade 12. I hope this video finds all students well from biology subject semester 2. I start with you today and we begin this talk today about inheritance changes. And first of all, let's start with last parts for the DNA molecules. First of all, DNA molecules is important part and the important point inside the nucleus. DNA molecules have more structures, phosphate groups, sugar, and also bases between the strands. And I give you an example to understand this point and start to answer after I make revision for this point. First one about the bases between the strands for DNA molecules. DNA helicase that have two strands that form phosphate sugars and bases between them. Bases, sequences of bases is very important point, and each one have counts or numbers of hydrogen bonds, the same. And start to understand each one, AT that have 2 hydrogen bonds and GC that have 3 hydrogen bonds. And start to understand the DNA helicase that hydrogen bonds that make attraction between strands to produce DNA helicase. This is about the last part, last lessons, and this is the first point to start today, that a new lesson for passage of information from parent to offspring. First of all, have more features for DNA molecules and have more details. Gene, coding strand, and also triplet code, and also that each one have one example. First of all, you must check this figure that DNA strands between bases between the DNA strands, ATGC that join the strands with together and make attractions between strands. But bases adenine, guanine, cytosine, thymines, all this have sequences that relatively short lengths. The first one, genes, that relatively short length of DNA that codes for single polypeptides. So to understand the short length of DNA, that understand the coding strand, that sequence of bases that found inside genes. Focus on this step by step. Strands of DNA molecules between bases, sequences of bases inside or between strands, that is gene. And gene is small length from DNA strands, small length from DNA molecules that have sequences of genes, and each one gene have coding strands that call sequences of bases. And coding strands have triplet code. Triplet code that mean is formed from 3 bases such as TAC, this first example. I give one example for the triplet code. TAC, that one strand that they have, you can say they have the opposite bases in the another strand. TAATGC, that you can say bases between the strands, the same, this part ATGC, all the sequences. Genes is small from strands, and inside gene have coding strands from 3 bases, ATGC, that sequences that produce coding triples, and the coding triples from 3 bases, ATG, for example, and each one from this triplet code that determined one kind from amino acid. So each one gene determined to produce one kind of polypeptides or one kind from amino acid to produce one kind of proteins. This about the small structure for the gene coding strand, triplet code, and also I give one example for this, that method. Let's move on another part for the organisms haploid and diploid. First of all, understand before in grade 10 or grade 11 that have more example about diploid and haploid. Diploid, that one cell that have 2 sets of chromosomes, and such as 46 chromosomes inside the human body. But haploid chromosomes, haploid cells, that one cell with 1 set of chromosomes, such as 23 chromosomes. And they give you structures for the chromosomes to understand that and make comparison between 2 cells with haploid and diploid chromosomes, such as homologous pairs. Lets us to make comparison between them, and after start to make features for chromosomes to check this figure and start to check all this point. Homologous chromosomes is that the two chromosomes that have the same position of centromere, the first one, that centromere, the first point, can say the major point between chromatids. Each one chromosomes from two chromatids, the same chromatids, and between two chromatids, that centromere, the major point that join with two chromosomes, and start all these chromosomes to join with together and have the same positions and also have the same position for genes. So homologous chromosomes that come together to, and you can say to specific pairs, to make specific pairs between them the same, this features between them the same characteristic, and also have more features for chromosomes the same. This number of chromosomes are fixed, such as inside the human body 46 chromosomes and 23 chromosomes in sperm and egg. And the shape of chromosomes the same I give you before, the same shape, the same length, and also the same centromere, and also the same position of genes. And number 3, that about gene and the alleles. It's very important point for features for chromosomes. Focus with me about genes and the alleles. Those features, you can say those feature of chromosomes. Listen to me about this point. Chromosomes, homologous chromosomes, and start to check the bases before I give you genes, that sequences, or can say a small length of DNA that have codes with 3 bases, can say genes, that sequences of bases that produce alleles. So genes, sequences that have coding, attributable to coding, and also that alleles form of genes, that A, B, B, this form, you can say alleles of gene sequence of bases. This about the homologous chromosomes and genes and also alleles. So I'll give one example for this. You must think about the questions and try to answer. Compare between alleles and gene. I give you the answers and give you more information when I explained these points about the gene sequences of bases and the alleles. Okay. This number 1 about the compared between alleles and genes. Number 2, start to do the process of meiosis. Meiosis, you can say reductive divisions, and what kind, this kind of process, this kind of cell divisions that produce gametes inside the human body, such as sperm and ovules, can say eggs, and start to produce gametes inside the flowering plants, such as pollen grains, and also egg inside the ovules, inside the ovary. This about the process that definition particular nuclear divisions. Because the process of meiosis, you must check the behavior of the nucleus, and special the can say the behavior of chromosomes inside the nucleus. This is a very important point, and I give you general shape about the process from meiosis, that one cell that start to have 2 divisions from meiosis number 1 that produce 2 cells, the first one, and start to make another cell divisions to produce 4 cells. Each one have half chromosomes from the first cell. So check with me about these stages to understand and make comparison between each stages. Let's move another part, the process of meiosis. Before I give you that example for stages of meiosis, have 2 stages, number 1, meiosis number 1 and meiosis number 2. So focus with me that each one from this have 4 phases, have 4 stages: prophase, metaphase, anaphase, and telophase. The same phases in the meiosis number 2, but the result is different from each other, that 2 cells, and the second divisions have 4 cells, that prophase number 2, metaphase number 2, anaphase number 2, and telophase number 2. And check with me all these stages and check the behavior. First one about the process of meiosis. First one about the first stage, prophase. Prophase can the first stage about the have more features because it's first stage. But I give you one point after and I explain this point, and they give you small notes about the prophase to understand the differences between prophase and metaphase. Before understand the chromosomes that can say shorten and thicken, that in the early stage before prophase, and start to condense with together to produce homologous chromosomes as the same. I explained before about the homologous chromosomes, same shape, same position of chromosomes, and same position of genes. So focus with me and check these points, homologous chromosomes. And after homologous chromosomes have one process, a very important point from crossing over, chiasma or chiasmata. That prophase can say that have more features. Number one, homologous chromosomes. Number two, that process is example for prophase number one, that is very important point for a huge genetic crossing over. That chiasmata, check this picture at the bottom of the page between the chromosomes and the chromatids. That you can say homologous chromosome, the same shape, the same length, and you can say the shape for the parts of the chromatids. And each chromatids start to make exchanges. You can say swapping species between each other. You can say between non-sister chromatids to produce a chiasma and to produce a different species in one point between non-sister chromatids. That you can say produce a huge genetic information inside this process. Let's move another part to understand this point, and I give you a small video about this point to understand process for metaphase, and it's very important point to complete this point. And the process of metaphase, the third phases, and you can say that have more features. At the end of the metaphase have more features such as spinal cord between the balls of the cells, and found centrioles that start to produce at the end of the prophase. And to check this point, the first one for this stages, you check this point about at the end of this stage start centrioles to produce at the balls, and the spinal cord that produce from balls to balls inside the cells, and the chromosomes start to lines up the equator of the cell, and you can say the middle of the cells, and then attached with spinal cord by centromere that join with a spinal cord and start to line up of the equator of the cell. Let's move another process to check this point, the telophase, and chromosomes start to separate from each other to one poles, each one from one poles and another one poles, and you must check the spinal cord start to break down and disappear. At the end of this phase, that number one, the nuclear envelope and nucleus start to reform to produce two cells, the same this figure, and start to produce, each one have the half chromosomes inside this. So the telophase is very important point from meiosis number one, that all chromosomes can say start to separate from each other to one pole. The same this figure, and you must check to understand the features for this. Number one, spinal cord start to appear. Number two, chromosomes start to separate, and centrioles start to disappear from the cells. And the last part start to produce two cells in anaphase. Homologous chromosomes start to separate as explained you before, and each one from these chromosomes start to produce nuclear envelope and start to produce nucleus again, and reform all these structures to produce two cells. Each one have half of chromosomes. This the meiosis number one. And let's move on another part for meiosis number two. That check this point, check this figure. That's one cell, and this meiosis number one, that homologous chromosomes that start to produce two cells, as explained with you about that. Inside this four phases, prophase, metaphase, telophase, anaphase, start to produce two cells, each one half chromosomes. After start meiosis number two, the same phases, prophase, metaphase, telophase, anaphase, but have more differences between them. That chromosomes start to divide into chromatids.